right, everyone, we're going to go ahead and get started. Thank you so much for being here today for our research seminar. My name is Cinnamon Moffett, and I'm the research program manager here at Oregon State University's Hatfield Marine Science Center. And you are at our research seminar this afternoon. This is a hybrid event. Most of you know that by now, but we have folks in the room and we have folks online. So welcome to you both. For folks online, we do have your cameras, mics, and screen shares disabled for this event, but please use the chat box if you have any technical issues or if you have any questions for today's speaker. We have somebody in the room that will help us moderate those, so feel free to use that chat box. For folks in the room, they're going to the very back row. Just raise your hand if you have a question and we'll bring you a mic so that you can ask that question and folks online can hear. But again, thank you so much for being here. If you weren't able to sign in on your way in for folks in the room, please do that. It helps us keep track of our outreach numbers for our speakers, but also helps us justify buying cookies and coffee for all of you. So again, thank you so much for being here. A couple of quick announcements. Next week, on August 1st, already is August, we have Alexandra Michito, as I'm probably saying that wrong, but we'll figure that out next week, is a collaborator with MMI that will be here to talk about environmental conditions in the rare human-dolphin mutualism. And this particular speaker will be remote, but will have on-site and remote viewing options. So I hope you come back and have another marine mammal talk. All of our recordings for our seminars are on our website. So if you go to our past seminar page, you'll be able to watch any of these recordings if you would like. But I'm just going to quickly get ready and hand it off to the folks that invited today's speaker. So Scott Baker and Lisa Balance are the ones that brought this particular person here and is helping to facilitate getting our speaker here today. So Lisa, I'm going to hand this off to you. I'm a little out of practice today, so.
Thank you, Cinnamon, and thank you all for being here. It's great to see so many people in person, also online. For those of you who don't know, I'm Lisa Balance, the very proud director of the Marine Mammal Institute here at Hatfield. And it's my great pleasure to introduce our science seminar speaker today, Dr. Franca Eichenberger. Franca, sorry, there I go. I asked her how to pronounce her first name. I've been saying Franca and been corrected to say that it's Franca. And she told me just before now that it's Franca, everyone. So Franca joined us here in the Marine Mammal Institute in March, just 5 short months ago. She's a postdoctoral scholar. She comes to us from the University of St. Andrews in Scotland, and she obtained her PhD there. Her original home is in Switzerland. So I can say, like her study animals that she's going to be talking to us about today, she is indeed highly migratory. Now, I had hoped to go into lots of detail about Franca's various accomplishments and expertise today in this introductory statement, but she has instructed me not to do so. She says she wants to share the story of her journey prior to OSU herself. So with that very short and simple introduction, I will hand the podium to her and let her tell us about finding fathers and remembering mothers using DNA profiles for long-term studies of humpback whales. Franca.
Thank you, Lisa, and welcome, everyone. So, yeah, I'm going to start with a very brief intro on, well, I guess my journey and how I ended up here in the US studying humpback whales when I grew up in landlocked little Switzerland on the very other side of this map. I did both my bachelor's and my master's at the University of Zurich in Switzerland. And for my master's thesis at the Department of Anthropology, I was looking at dispersal strategies and male reproductive success in wild vervet monkeys. And as part of that, I went to South Africa to conduct field work on wild rabbit monkeys for half a year in order to collect both behavioral and genetic data. And what I really mean with genetic data is poop. And after all the lab work and staring at the computer screen during the finishing of my thesis, all I wanted to do was go back into the field. So I joined the elephant seal research group on the Falkland Islands for a few months to collect again behavioral data on both elephant seals and killer whales, which was really my first experience or close encounter with marine mammals. And I then went on to do a PhD at the University of St Andrews in Scotland, which was also focused on male reproductive success, but this time on a humpback whale population in New Caledonia in the South Pacific, and many roller coasters, a global pandemic, and a short excursion into the world of Hawaiian field crickets later, I finally defended my thesis at the beginning of this year. And here I am as a postdoc at MMI OSU, still looking at genetic data on humpback whales, but in the North Pacific this time, basically right at our doorstep. And I'm part of the Cetacean Conservation and Genomics Lab, led by Scott Baker and generously funded by the Gray Whale license plate. And in this talk, I will mainly focus on some of the work I did during my PhD at the University of St Andrews, but I will also provide an outlook or overview of what I'm currently working on here at OSU. And both of these analyses make use of long-term genetic sampling on humpback whales. So I just want to kind of show you some of the many, many different things that can be done or analyzed with genetic data, especially if collected over long periods of time. So in the first part of the talk, we'll find fathers to assess patterns of paternity on a humpback whale breeding grounds. And in the second and shorter part of this talk, we will remember mothers and assess patterns of genetic diversity resulting from maternal fidelity to feeding grounds. So, but before I dive into any of this, let me introduce you to both humpback whales and DNA profiles. Humpback whales can get up to 18 meters in length. Females are slightly bigger than males. And they have these massive pectorals that can be up to 5 meters long. That makes them look quite different compared to other baleen whale species. And that's also what's giving them their name. Megaptera means giant wing or big winged. And from the perspective or coming from primatology, they really look a little bit like the gibbons of the sea. And they are estimated to reach up to 90 years of age. And humpback whales, like many other baleen whales, are capital breeders. So this means that they rely on abundant prey resources to fuel their annual migration and reproduction. They fill up their energy reserves on their feeding grounds at high latitude during the summer months and reproduce at their low latitude breeding grounds during the winter months. And that's the case for humpback whales in the northern and southern hemisphere, just that the winter and summer months are reversed, so they don't really get to meet. They show maternal fidelity to feeding grounds and natal fidelity to breeding grounds. So they tend to go back to breed where they were born, and they return to feed to where their mothers brought them during their first year of life. So maternal traditions learned during their first year of life. And humpback whales across the world have a long history of human exploitation. Commercial and sometimes illegal whaling lasted deep into the 20th century, and humpback whales across all oceans were decimated to very low numbers. The level of their recovery varies considerably across populations today. Some populations are considered to have fully recovered, while others are still considered endangered. And on a more brighter side, we can identify individuals based on photo ID of their fluke and based on genetic IDs derived from DNA profiles. But how do you get from a humpback whale to its DNA profile? Biopsy samples of humpback whales are typically collected using a crossbow or a modified veterinary rifle. And this usually gives us both a piece of blubber and a piece of skin. And we can then extract DNA from the skin in the lab to get both nuclear DNA, which is biparentally inherited, and mitochondrial or empty DNA, which is inherited from the mother only. The extracted nuclear DNA can then be used to determine the sex of an individual, and we can create multi-locus microsatellite genotypes for individual identification. Using mtDNA, we can determine the mtDNA control region haplotype of each individual. So DNA profiles can be constructed using these three things: sex, microsatellite genotype, and mtDNA haplotype. And an extensive database of such DNA profiles of many individuals collected over many years or even decades is often referred to as a DNA register. And today I will talk about two of such DNA registers and give example of what kind of analysis and questions can be tackled with DNA registers in humpback whales. So let's start. In New Caledonia, an island in the South Pacific, somewhere between Australia and New Zealand, just a bit more north. And the New Caledonian humpback whale breeding ground is really part of the wider Oceanian metapopulation, which today is still considered endangered under the IUCN Red List. And compared to feeding grounds, humpback whale breeding grounds are characterized by an elongated fasting period of individuals. As I mentioned, they are capital breeders. They're also characterized by a male bias sex ratio. So this means that there are a lot more males than females on the breeding ground, which leads to intense competition among those males over those females in order to reproduce. And on these breeding grounds, males frequently engage in a variety of different mating behaviors. They physically compete over access to a single mature female in so-called competitive groups of 3 or more individuals. And within these groups, males often engage in agonistic fights to gain and maintain the position closest to the female of the group. They are also frequently observed escorting a single mature female or a mother with calf. And we don't really know if escorting leads to the formation of competitive groups, or if an escorting male is the winner of a competitive group as he managed to outcompete all of his competitors. Or in other words, it is unclear whether escorting a female results in copulation or whether it reflects mate guarding following earlier copulation. Also, mating in humpback whales has never or hardly ever been observed. And last but not least, humpback whales sing a complex vocal display. And the function of their song is still debated, but it has been suggested that song serves a sexual function and so might act as some sort of acoustic peacock tail, with sexual selection as the driver of this song complexity. And my PhD supervisor, Ellen Garland, has a project called Sexy Singing Project that seeks to explore these underlying selective forces interacting and governing various aspects of humpback whale song. And my thesis was part of this wider project, but I didn't look at song. I focused on patterns of paternity and male reproductive tactics to increase our understanding of the possible selective forces that could be acting on these behaviors. And I'm very grateful for the support of this really amazing team of supervisors, Ellen Garland and Luke Randall from the University of St. Andrews, Emma Carroll from the University of Auckland, and Claire Garrigue from the IRD in Entropy in New Caledonia, and Opération Cétacé, which led the fieldwork in New Caledonia. So, sexual selection. Let's start with a definition. Sexual selection is any selection that arises from fitness differences associated with non-random success in the competition for access to gametes for fertilization. In simple words, and in the context of male humpback whales, when some males are more successful in reproducing than others, this leads to variation in reproductive success. Some males might be more successful than others because they are, for example, superior in strength, stamina, or they are maybe more attractive to females, or perhaps some are better singers than others. And so within that definition, there is or there might be a correlation between these two sides. But humpback whales spend all their life on the water. And mammals that mate on the water tend to show lower variation in male reproductive success. Here we are looking at the standardized variance in male reproductive success across a range of species and taxa. And the first thing that becomes pretty obvious is that that variance in male reproductive success is considerably lower in species that mate in the water. Baleen whales, with southern right whales, humpback whales, and North Atlantic right whales, Galapagos sea lions, killer whales, and bottlenose dolphins all show relatively low variance in male reproductive success. Whereas mammals that reproduce on land, such as vervet monkeys, red deer, rhesus macaques, and at the very top, elephant seals with their harem mating system show a much larger variation in reproductive success. Yes, elephant seals are marine mammals, but they are mating on land. And so the three-dimensional underwater habitat, which makes controlling access to mates a lot more difficult than it is on land, together with the wide distribution of individuals across the breeding ground or oceans, serve as very likely explanation for this reduced variation in reproductive success in aquatically mating species. But despite this expected low variation in male reproductive success, we don't really know whether some male humpback whales are more successful than others. And if so, what exactly makes one male more successful than another? As not many studies have looked into patterns of reproductive success in baleen whales in general. So for the first part of my PhD, I looked at patterns of male reproductive success in humpback whales. And specifically, I assessed the variation in male reproductive success and looked at non-random patterns. And as a next step, I then looked at the role of age in shaping patterns of male reproductive success. And that's what I'm going to talk today. And looking at these questions was really only possible thanks to the long-term monitoring of the New Caledonian humpback whale breeding population, which was led by Opération Cétacé. And survey efforts were focused in the South Lagoon, so the southern end of the main island, but shallow reefs close by were surveyed as well. And surveys started in 1995 and are still ongoing. Here I looked at data collected until 2020, so a 26-year-long data set. And these surveys were conducted during the breeding season in Austral winter, so that's July to September. And I was very lucky to have had the chance to join Opération Cétacé in this amazing team for 2 field seasons. And just to give you a little glimpse into how fieldwork looked like, we usually filled the boat around sunrise, seeing how bright it is in this picture. I think we had a bit of a late start on that day, but so we then had usually a team at sea that was in constant contact with a team that was based on land. And that land-based team then usually got dropped off along the way and then had to hike on top of this hill to help spotting whales from the lighthouse. And the soil was very rich in iron oxide, which gives it this very orange color, so that by the end of the day, the land-based team was always the one with the orange feet. So during those yearly field surveys, we collected data on the behavior, photo ID, biopsy, and song recordings of individually identified humpback whales. And all of this effort led to this unique data set of almost 6,000 observations, almost 3,000 biopsies, 100 song recordings, and over 1,600 genotyped individuals at 15 microsatellite loci, of which most were males, which mirrors that typical male bias sex ratio observed on humpback whale breeding grounds. So I used those microsatellite genotypes to find fathers. I did this by conducting a paternity analysis on 177 known mother-offspring pairs, representing about 55% of all mother-offspring pairs observed since the start of the first survey in 1995, and a bit more than 900 mature males. And to give you a very, very brief overview of how that works in principle, we have our known mother-offspring pair, and we know their genotype. We know that the offspring inherits one allele from the mother and one from the father. So we can then derive from that which allele the offspring must have gotten from its father. And we can then check whether any of our sampled males could be the possible father following Mendelian inheritance. But there might be multiple possible candidate fathers. But because we're not looking at one locus, but 10 to 16 loci, so imagine 10 to 16 of those colored bubbles for each individual, which then gives us a much better resolution to find those fathers in our sampled males. And I did this in a software called Service, which implements a pairwise maximum likelihood method to assign these paternities. And I was able to assign 66 males as fathers to 79 of the sampled offspring. And as you can see in the graph on the right, most sampled males didn't sire any of the offspring that we sampled. But it doesn't necessarily mean that none of those 870 males were not able to sire offspring, but that they most likely didn't sire any of the offspring that we were able to sample. So although this is strongly influenced by the proportion of individuals sampled, the fact that we sampled over half of the offspring that we observed still suggests that most males were not able to sire offspring over the course of the 26-year-long study period. So it was skewed towards males that did not sire offspring. Focusing on the males that did sire offspring, we can then see that no male in our data set sired more than 2 offspring over the course of the study period, and that there were only 1.2 offspring per father. And so as expected, variation in male reproductive success in humpback whales is low. And it was as low as it is in other baleen whale species. And variation in male reproductive success of our humpback whale breeding population in the South Pacific was very similar to that one other paternity study on humpback whales in a population in the North Pacific. Okay, so as expected, variation is low, but is it random? Or are some males more likely to reproduce or more successful than others? Because low variation doesn't mean random and doesn't mean that each male is equally likely to sire. So to test whether some males are more successful than others, I conducted a simulation analysis to assess whether the observed number of paternities deviates from the distribution of paternities expected under the assumption of random mating. And as we've seen earlier, based on the number of sampled offspring, most males are expected here in black not to have sired any of the sampled offspring at all. And because this number is so much bigger, for the number of males that did sire, I have scaled another y-axis on the right. And the expected distribution of reproductive success derived from these random mating simulations, shown in white, differed significantly from the observed distribution of reproductive success based on the paternity analysis, shown in black. And this was mainly due to fewer males than expected siring one offspring and an excess of males siring two offspring in the observed distribution compared to the expected distribution. So male reproductive success does not appear to be random, raising the question, what makes some males more successful than others? So as a next step, we then wanted to explore the role of age in shaping these patterns of male reproductive success. And to estimate the age of individuals, we used a tool called epigenetic aging. And for anyone who now tries to remember what epigenetics is again, let me refresh your memory. Epi means on or above in Greek, and epigenetics describes factors beyond the genetic code. So very simply put, epigenetic changes are modifications to the DNA that regulate whether genes are turned on and off or off, but they don't change the DNA sequence itself. And DNA methylation is one epigenetic mechanism during which a methyl group is attached to the DNA. And so epigenetic aging makes use of the correlation between DNA methylation and age at specific DNA regions. And for this, we collaborated with Simon Jarman, who was part of the earlier study by Polinovsky et al. In 2014 that developed the so-called epigenetic age assay for humpback whales. So we sent a bunch of samples to the Australian Genome Research Facility that measured the DNA methylation levels of 8 specific regions in the genome that were previously found to show a significant age methylation relationship in humpback whales. And I'm not going into any more details in the aging methods here, simply for the sake of time, but please reach out if you do have questions. So with that, we were able to age around 485 males. And comparing the estimated age and the known age of individuals that were observed as calves within the study period showed that we were able to estimate the age of individuals with an accuracy of ±5 years. And so because we don't really have the accuracy for exact point estimates, we then decided to group individuals into different age categories that were based on these measures of uncertainty and the age and the estimated age of sexual maturity in humpback whales, which is around 9 to 11 years. And calves and yearlings were excluded from the analysis. The oldest age category was left open-ended as we were unable to measure uncertainty for age estimates older than the length of our study period. And then there was a big increase in the New Caledonian humpback whale breeding population around the year 2008. And this nicely divides the study period into 2 time windows, one of lower abundance and one of higher abundance. So because of this, we expect more and more older animals in the second time window. So I then assessed age-dependent male reproductive success within each of those 2 time windows separately. For that, I conducted another simulation analysis where I randomly selected the number of fathers from the underlying population age structure, again under the assumption that each male has equal chances to reproduce. I then compared the simulated number of fathers with the observed number of fathers from the paternity analysis to assess whether males of a particular age category were more or less likely to reproduce based on this underlying population age structure. So, for example, here, in basically none of the simulations was the observed number of fathers larger than the expected number of fathers. Or in other words, 2 to 9-year-old males were less likely to sire than expected based on the underlying population age structure. The age of sexual maturity in humpback whales was estimated to be between 9 and 11 years. So this is what we would expect. And with just a quick look at the observed age structure of fathers from the paternity analysis, we can see that this looks quite different across the two different time windows. And just by looking at the expected number of fathers from the simulations that are based on the underlying population age structure, we can also see that there was a shift in the population age structure from a population that was more skewed towards very young individuals in the first half of the study period, closer to the end of commercial whaling, towards a more balanced population age structure in the second half of the study period. And now looking at how the observed age structure compares to the expected age structure of fathers based on the underlying population age structure, and assuming that each male, regardless of age, has equal chances to sire offspring. So because there aren't many older males in the first half of the study period, we also didn't find or sample any of the oldest males that sired offspring in our data set in the first time window. But males between 9 to 16 and 16 to 23, here in red and blue, were overrepresented compared to these simulations. And in the second time window, when the population age structure was more balanced, 9 to 16 and 16 to 23-year-olds were no longer overrepresented, and we suggest that they had their paternity rates reduced by an increase in the presence of those older males in the second half of the study period. So this indicates that age does play a role in shaping these patterns of paternity in humpback whales, but that the age structure of fathers also depends on the underlying population age structure. So let's recap a few main points. We were able to confirm the low variation in male reproductive success, but also showed that it's not random, that some males are more successful than others, and we found indications that reproductive success in male humpback whales is age dependent, but that these patterns of paternity are also conditional on the underlying population age structure. We also saw that there was and is a shift in the population towards a more balanced age structure, indicating signs of population recovery. And that whaling or exploitation in general likely has long-lasting impact that goes way beyond the number of individuals in a population, which highlights the importance of long-term population monitoring in order to assess these evolutionary consequences and changes across time, especially in species with long lifespans. And lastly, that we have to be aware of shifting baselines. What is normal today may not have been normal before or pre-whaling, and what we see today is a snapshot in time and needs to be put in the right context. What we observe today may not reflect the conditions under which certain behaviors have evolved. So far, I've talked to you about how we can use microsatellite genotypes to analyze paternity and to assess patterns of male reproductive success. Now we leave the humpback whale breeding ground in the South Pacific and return to the US West Coast in the North Pacific. And this time, we focus on a different part of that DNA profile, the maternally inherited mtDNA haplotype. At the beginning of this talk, I told you how humpback whales undertake seasonal migrations to feed, mate, and give birth. We also know that they show natal fidelity or natal phallopatry to breeding grounds and maternal fidelity to feeding grounds, which are maternal traditions learned during their first year of life. but they not only inherit their migratory destination from their mother, but also the mtDNA haplotype. So this site fidelity is really reflected in the genetic structure of populations, and the distribution of mtDNA diversity or composition reflects the stability of maternal fidelity over generations on an evolutionary timescale. But it's not quite as simple or straightforward as this map might suggest. There are varying degrees of mixing between feeding and breeding grounds. Individuals can share fidelity to one breeding ground, but not to the same feeding grounds or vice versa. And because of this asymmetrical pattern of interchange between seasonal habitats, there are no simple divisions of populations into migratory subpopulations. So the seasonal migrations of these population units or herds creates a complex spatio-temporal and genetic structure with challenging implications for management. So, but why is assessing the genetic structure of these populations important? For example, whales are at risk of ship strikes and entanglement in fishing gear, and these risks vary along the coast. There are also more global issues such as climate change and heat waves that, while often affecting a large area, can fall out more intensely in one area than another. Overall, this leads to varying risks of injury, death, or lack of sufficient food resources along the coast and across years for humpback whales and other species. And such issues on feeding grounds will directly impact their respective populations on the breeding ground through, for example, death or reduced reproductive rates. And humpback whale populations across the North Pacific show varying levels of recovery from commercial whaling. While the Hawaiian population is still considered fully recovered, other breeding grounds in Mexico or Central America are still considered threatened or even endangered. So depending on which breeding ground these individuals on impacted feeding grounds along the West Coast return to for their reproduction can have more or less detrimental effects to their breeding grounds or breeding populations overall. Also, local exploitation can not only result in the loss of genetic diversity, but also the loss of the basis for the inheritance of migratory destinations. So how can we use the DNA register to better understand what's going on and how and which whales are feeding along what parts of the coast, and how strong is this site fidelity? How faithful are they to these maternal traditions? Over the past 3 decades or even longer, a lot of biopsies from humpback whales have been collected along the US and Canadian West Coast. And in this part of the talk, I won't really give you any results, but I'll provide you with an overview of how such a DNA register can be used to look at the genetic population structure and genetic diversity of humpback whales, focusing on some of the key points that I'm currently working on here at MMI. So we have, starting from the very northern end of this map, Southeast Alaska and British Columbia divided into northern British Columbia and southern British Columbia, Washington, Oregon, and California divided into North, Central, and South California, to create somewhat even regions along the West Coast that largely follow political and state boundaries. And in the case of BC and California, some landmarks that often correlate with gaps in the sample collection. And the number of collected samples varies considerably across regions, as some regions were surveyed and sampled more intensely. And some of these more intensely sampled regions, such as Southeast Alaska and Central California, have been surveyed over many years. So that's allowing to not only look at changes in the genetic diversity across space, but also across time. And so within a population or a region, we then often look at the genetic composition. So for example, the frequency of mtDNA haplotypes, which are then often visualized as pies. And get ready, because there are 24 more pies coming for you. So if we look at the mtDNA haplotype frequencies of these 8 regions along the West Coast, we can see that some regions show a higher number of different haplotypes than others. So what can we look at with this kind of data? We can assess the fine-scale spatial genetic diversity and how stable this is across time. And we can start looking at which breeding ground these individuals might be coming from. So looking at this genetic diversity across space or latitude, to be more specific, we can already detect a clear pattern across regions with more yellow or A- in the north and more green and blue, so E and F type haplotypes, the further south we go. And at the bottom graph here, we're just looking at the 4 most common haplotypes, so A-, A+, E1, and F2, and we can see a clearly clinal pattern of mtDNA haplotypes along the West Coast. But if we zoom in even closer into Oregon, for example, and look at an even finer scale, we can find that same pattern. More yellow A- further north, and more green and blue E1 and F2 further south along the Oregon coast. And for regions with a large number of samples collected across many years, we can then start looking at how stable these pies look like across different time blocks that are based on the number of samples collected to ensure a big enough sample size. And so for Southeast Alaska, we can see that the frequency of mtDNA haplotypes appear very stable across this almost 30-year-long time period. And for Central California, the pies also look very similar across years. They look more similar across years within Central California than across adjacent regions. But we can also start seeing more yellow or A- popping up in those more recent years. So does that mean that A- individuals that are usually more common further up north now start venturing further down south? Or are these A- individuals simply migrating along the coast, aiming for their feeding grounds further up north? Or is the proportion of A- individuals on their respective breeding grounds increasing? There are several possible explanations, but what is clear is that even if there might be small variations to this structure across years, the overall genetic composition shows strong temporal stability, which really highlights the. And lastly, and because of this, we can then start looking which regions and how well regions match the DNA haplotype composition of breeding grounds. And to some degree, we can see that same clinal pattern of mtDNA haplotype composition or frequency along the US and Canadian West Coast mirrored across humpback whale breeding grounds in the eastern North Pacific. So that was an overview of some of the things that I'm currently working on in the form of a lot of pies. And I'm just going to leave you with 2 take-home messages. First, long-term population monitoring and sampling is important to assess patterns of paternity in long-lived species, such as the humpback whale, and to assess the genetic population structure and population dynamics at an evolutionary timescale. Also, we should keep in mind that biopsying is considered minimally invasive, and that these samples are highly valuable in that a lot can be learned from a single sample. This really highlights the importance of collaborative datasets that can lead to big DNA registers, especially for such highly migratory species like the humpback whale with their complex spatiotemporal structure. And both of these points become even more important in light of shifting baselines as whales keep recovering from commercial whaling over time, or start to shift from recovery to climate response. And that brings me to my acknowledgement slide, which even further highlights just how big and how wide-ranging this effort is. For example, within MMI, to collect samples in the North Pacific, and also within MMI, and especially the Cetacean Conservation and Genomics Lab with Scott Baker and Debbie Steele, that played and are still playing a major role in curating these DNA registers and bringing all these samples together into one place for humpback whales in both the North and South Pacific. So thanks to everyone that helped creating these DNA registers, and thank you for listening.
Thank you so much, Franca. Appreciate that. All right, we're going to bounce back and forth with questions. Katie, do we have any questions online? Okay, for folks online, if you have any questions for today's speaker, go ahead and put them into the chat. How about questions in the room? One moment, please.
Thanks, Franca. Amazing work. Really beautifully told to see it all come together. My question is actually about the first part and the paternity analysis. It was interesting to hear that your metric of within your data set, I think you said 1 to 2 offspring per father that you were able to identify. And I'm curious how that compares to if you have a similar number for known mothers, either from biopsy or from photo ID, whether sort of within your data set, how many offspring the moms tend to have.
Yeah, there were females of those non-mother offspring pairs that sired up to 4 offspring. So it was, yeah, a lot bigger. But I say 1 to 2 offspring per male, but like up to 2 offspring within the study period. But we also need to keep in mind that this is only a short kind of snapshot of their lifespan, which they can get up to 90 years old. So it could be a bit more or quite a bit more than just the 2 offspring. Yeah.
All right, other questions in the room?
Yeah, great talk. I really enjoyed that. My question is about that ratio between males to females in New Caledonia with 1.5 males to 1 female. Where are the other females, I guess, is my question. I mean, they must go someplace, or is the whole population like that? And do you see a similar ratio on the feeding grounds here in North Pacific?
Good question. On the feeding grounds, it tends to be more even. So those females have to be somewhere, but they might. One theory is that not all females migrate because it's very energetically demanding to reproduce, and they don't reproduce every year. So maybe they just decide, I'm gonna skip this year, I'm gonna continue eating and maybe go next year. So that's kind of, it could be a kind of reproductive tactics of females to decide, am I ready to reproduce and go on migration and then not eat on the breeding ground, or maybe next year. Yeah.
All right, other questions in the room? I'm just gonna stay right here for a sec.
Hi, thank you. That was an excellent talk. This is probably basic, but as someone who doesn't know so much about mitochondrial haplotypes, I was wondering if the A haplotype variants or the B's or the E's, because you put them in similar colors, are they more similar to each other? And does that have any meaning?
Yes, yes. So they have a different number of base pair differences, but the F ones are usually more similar to each other, and the E ones and the A ones. And I could show you a kind of tree or kind of a plot that shows the differences between those. But yes, that's well spotted.
All right, there's a question at the end of this row. Can you hand the mic down?
Hi, Franca. Thank you so much for the awesome presentation. But I was wondering if you investigated, and if you did you find any correlation between the male reproduction success and the bioacoustic, the songs? Did you investigate it?
Yes. So that was kind of I mentioned the Sexy Singing Project. So that's the big aim of that project. And my PhD was kind of setting the foundations. And there's now people that take that and continue looking at various aspects of song. They look at the correlation between reproductive success and song complexity, but also novelty. So that's ongoing work, and hopefully we'll know more soon.
Thank you. Thanks.
Hi, Franca. Excellent presentation and amazing work. I can believe that we share office and I didn't know about your research. But just maybe you can clarify to me like about the feeding areas. So different haplotypes feed in specific areas. Is that correct? Makes sense?
That would be very nice, but it's a bit more it's a bit more mixed. There are some haplotypes that are more common, like the A-1 that occurs predominantly or more often or more commonly in the north, but also a little bit further south we go. So there are some haplotypes that are more frequent in some areas along the coast, but we can't really say they just occur there and they just occur in that breeding ground because of this very asymmetrical mixing of population units or migratory units. But we're trying to see both together with mtDNA haplotypes, but also trying to include the microsatellite genotypes that maybe help us find out how individuals move and which individuals from which breeding ground go to which areas along the coast.
Yeah, the reason why I'm asking is because if you combine with stable isotopes, with Kelly and I, we tested this for the sperm whales and we saw that different haplotypes consume different prey sources. So maybe if you combine these techniques, you can see like haplotypes from a different feeding ground, or these haplotypes are eating in specific feeding grounds. So maybe can help you focus in a specific area.
Yeah, that is an excellent idea. If we could look at how that signature looks like along the feeding ground and then compare it with, I don't know how long you can measure those stable isotopes, if you could still kind of detect a signal when they're on the breeding ground and not eat. Yeah, I mean, that would be such a cool study.
Yeah, I think you can. We can talk
Okay,
more.
let's
But
All right, any other questions in the room? Okay, I'm going to get there. Any other questions online, Katie? Okay, we'll process it. Okay, we'll get one up here.
Hi, Franca. That was an amazing talk. I wondered for the study of the male reproductive success, did you have any other metrics about the individuals, other than obviously looking at song, but their size or their like blubber thickness to indicate their body condition or their health that would indicate some other advantage in competition?
We did not have info on size or body condition because we didn't have a drone pilot.
Hint, hint. I was really interested in your graphic you put up at the end about how the stability of the mitochondrial haplotypes over time in Southeast Alaska and in California. That was really interesting to see. And you alluded to the question about sampling within over months of a year, over year and cycles, and to try and capture, migratory versus foraging animals in certain geographic areas. So have you looked at that, or do you plan to look at that? Basically the same sort of plot, but by month instead of by year?
Yes, I did look at that. I think only in Oregon for now. But it kind of cuts up the sample size into even smaller pieces. So it makes it a little bit harder. Yeah, but it is an interesting question. I need to see if there are enough samples to look at that. Yeah. Yeah. Yeah.
All right, got a question online. Go ahead, Katie.
Okay. Have you seen any evidence of sampling bias contributing to the uneven sex ratio on the breeding grounds? In other words, are females harder to approach and sample on breeding grounds than males?
Good question. Female with calves are, yes, they are harder to approach. But I didn't have the feeling that females in general were harder to approach, but also when we're on the boat at sea, we don't know if it's a male or female unless it's a bit obvious in a competitive group or unless there is a calf. So that would be an interesting study, I guess, but I guess we wouldn't be able to say because we have approached if we have a sample. But I think another important point is that the breeding ground is also quite a large area, and our survey effort was mainly focused in the South Lagoon. And there were quite a few mothering calves, and they tend to be more kind of inshore than the males. But there are also other kind of sea mounts close by, and it's just impossible to survey all those areas at the same time. So there could be more females somewhere else that we didn't see. But the sex bias, male bias sex ratio on the breeding ground is really a common thing that is observed on like all humpback whale breeding grounds, not just the New Caledonian one.
All right, we have run against the end of our time, so can we give Franca one more round of applause? Thank you so very much, Franca, and thank you everyone for being here. We'll see you next week.